Adalings, in this video we are going to discuss about the cell division. Firstly, the cell cycle. The cell cycle is completed in two phases. One is interphase and M phase. Interphase is also known as the resting phase because in this phase the cell won't divide. It will prepares for the M phase. M phase or division phase. So this is also known as the resting phase or preparation phase because it prepares for the next phase, that is mitotic phase. This interphase is divided into three subphases. One is G1 phase or gap 1 phase, S phase or synthetic phase, and G2 phase or gap 2 phase. And after G2 phase, it leads to the M phase or mitotic phase. In this mitotic phase, the cell division takes place. This M phase is again divided into two phases. One is karyokinesis and cytokinesis. Karyo means nucleus, kinesis means division. Means karyokinesis means nucleus division takes place. Under cytoplasm kinesis. Cytokinesis means cytoplasm division takes place in this cytokinesis. This karyokinesis is again divided into four subphases. One is prophase, metaphase, anaphase, and telophase. This is the cell cycle. It mainly starts with the G1 phase. In G1 phase, cell won't divide. It will prepares for the S phase. In this G1 phase, RNA synthesis increases and ATP synthesis also increases, and size of the cell increases, and the raw materials or enzymes which is required for the S phase will develop in this G1 phase. The protein synthesis also increases in the G1 phase. Mainly the enzymes which is required for DNA replication takes place in the S phase, and the enzymes which are required for this DNA replication will synthesize in this G1 phase. After G1 phase, it leads to the S phase or synthetic phase. In this phase, the DNA replication takes place, and as well as the RNA synthesis increases and the histone protein synthesis increases in this phase. And after S phase, it leads to the G2 phase. In G2 phase, both same as in G1 phase. In this phase also, the ATP and RNA synthesis increases, and size of the cell will keep on increasing from this phases to phases. In this G2 phase, the protein synthesis also increases. Mainly the tubulin protein which is required for the spindle fiber formation will increases in this G2 phase. After G2 phase, it leads to the M phase. In M phase, it is actual cell division takes place, and it is divided into four subzones. One is prophase, metaphase, anaphase, and telophase. This we will discuss later. And after M phase, after cell divides, it leads to the cytokinesis. This is also in the M phase. All these are the karyokinesis, means nuclear division takes place. After this nuclear division, it leads to the cytokinesis, where the cytoplasm division takes place. After this cytokinesis, two daughter cells are developed. These daughter cells, some cells enter into the G1 phase for division. In the middle of the G1 phase, they won't divide or develop further, and leads to the G2 phase. This G2 phase actually comes in between G1 phase. We written here, but it will come in between this G1 phase. The cell will grow for some time, and it will lose the ability to divide further, and leads to the G2 phase. In this G0 phase, we will see de-differentiation, differentiation, and re-differentiation phases. Differentiation means the cells have lost the capacity to divide, and they are matured, to perform a specific function, and they won't divide further. Such type of cells are known as the differentiation cells, and when required, the capacity of the division of the cell will last. When they required, they again regain the capacity of the dividing cells to divide the cells, and this process is known as the de-differentiation. And all this differentiation, de-differentiation, and re-differentiation will seen in this G0 phase. In this cycle, the chromosome number remains same, but the C value will become double in this S phase. C value means the total DNA content in one set of the chromosome. And in firstly in G1 phase, the chromosome will be diploid, and the C value will be the 2C. 2C because diploid means it contains 2C means C value means the total DNA content in one set of the chromosomes. Here two sets of DNA is there, so the C value is also double, 2C. And when it comes to the S phase, here the DNA replication takes place. See, this is the chromosome, and it has both parents chromosomes. See, this is one chromosome, and it has both parents DNA content. And in S phase, the chromosome remains same, but the DNA undergoes replication and leads to this structure, this type of structure. Here the DNA replication takes place. So the C value, the chromosome structure remains same, but the C value increases. So the chromosome structure is 1, but the C value leads to the 2, double, it will become double. So after a space the chromosome will be deployed, but the C value become double and become to the 4C, and it will continue towards this way, towards the cycle. Mitosis. Mitos means thread and osis means state. Stransberger is the scientist explained the mitosis process in the plant cell, and Fleming is the another scientist who explained this process of mitosis in the animal cell. And mitosis mainly takes place in the stomatic cells, both haploid and diploid, and it also takes place in the germinal cells during the maturation phase. This mitosis is divided into two phases. One is karyokinesis and cytokinesis. Karyokinesis is again divided into 4 subphases. One is prophase, metaphase, anaphase, and telophase. Karyo means nucleus, kinesis means the division. Cytokinesis, cyto means cytoplasm, and kinesis is the division of this cytoplasm. In prophase, again you can see early prophase and late prophase. In the early prophase, the nuclear membrane, the endoplasmic reticulum, the Golgi nucleus, which is attached to this nucleus, will disassociates or become fragments, or starts to degenerate, and the centrioles, which is near to this nucleus, will present towards this nuclear, now migrates towards the opposite pole. One remains towards the one pole and another comes to the another pole, and this chromatin fibers condense to form the chromosomes. This centrioles is present only in the animal cell, and in the plant cell we cannot see this centrioles. In the late prophase, from this centrioles these structures are developed. Some fibers are developed, known as, one is small fibers, another one is the long fibers, and around these centrioles the fibers, which is short fibers, are known as the castor rays, and along with this centriole and the castor rays together known as the aster rays. Both these castor rays and centriole together known as the aster rays. In the late prophase, the spindle fiber formation is completely takes place, and in the animal cell these asters are two. Aster rays are the castor, the rays around this centriole is known as the castor rays. Along with this castor rays, the centriole is together known as the aster rays. In the animal cell, two aster rays are present, so these animal cells are known as the amphiastral cells, and in case of plant cell these aster cells are completely absent, so they are known as the anastral cells. At the same time, this spindle fiber contracts and the chromosomes comes to the middle of this. Middle means middle to this spindle fiber, middle to the spindle fibers. After late prophase, it leads to the metaphase. In the metaphase, these chromosomes align on the equator plane, means these are at the middle of this spindle fiber formation, and the spindle fibers are attached to this kinetochore of the chromosomes. And two types of spindle fibers are seen. One is continuous, where the chromosomes are not attached to this spindle fibers, and such type are known as the continuous fibers, and the chromosomes which is attached to this spindle fibers, and such type of fibers are known as the discontinuous fibers. This continuous fibers is also known as the interpolar fibers, because it runs from one pole to the another pole, and this discontinuous spindle fibers or discontinuous fibers are known as the chromosomal spindle fibers, because this spindle fibers contains the chromosomes, so these are known as the chromosomal spindle fibers. This metaphase leads to the anaphase. In this anaphase, this spindle fiber starts pulling these chromosomes towards the pole, and this is the structure of the chromosome. In the G1 phase, after the S phase, in the S phase of the interzone of the interphase, the DNA replication takes place, and the structure of the chromosome changes to the like this. We cannot write everywhere like this, so we will write like this. This is the actual structure of the chromosome after the S phase. Now this spindle fibers, which is attached to this kinetochore, starts pulling towards the opposite, towards their poles. Because of this spindle fibers pulling these chromosomes towards the pole, the interzonal fiber is formed. See, this centromere starts dividing because of pulling this spindle fiber. The centromere divides and splits in the anaphase. Because of this splitting of this centromere, see, one layer moves to the one pole and the another layer moves to the another pole. Because of this, the interzonal fiber is developed in the anaphase. Due to pulling of these spindle fibers, the shape of these chromosomes changes, and different types of chromosomes, different shapes of chromosomes is seen only in the anaphase because of pulling of these spindle fibers towards their poles. And the shapes are K, L, J and M shapes. K shape is metacentric, where the centromere is at the center and the two arms are of equal size. And the L shape is the submetacentric. In this, one arm is somewhat short and another arm is somewhat long. And J shape is the acrocentric. In this, one arm is completely short and another arm is completely long. And M shape is telos M shape is the telocentric, where this only one arm is seen. These shapes are due to the pulling of these spindle fibers. This pulling of spindle fibers, this chromosomes shape changes because this spindle fibers starts pulling these chromosomes towards their poles. And the forces responsible for pulling these chromosomes towards their pole are stretching of interzonal fibers and contraction of this chromosome spindle fibers and the ATPs. All these forces helps in pulling these chromosomes towards their poles. After this anaphase, these chromosomes will move towards their poles and this interzonal fiber is degenerated, and this anaphase leads to the telophase. This telophase is reverse to the prophase or early prophase, because in the early prophase, the nuclear membrane, endoplasmic reticulum, Golgi body all disappears or degenerates. But now in the telophase, the nuclear membrane reappears and the chromosome condenses to form the chromatin fibers, and the asterase and spindle fibers will degenerate, and all the membranes like Golgi complex, endoplasmic reticulum will starts reappearing. This is the karyokinesis, means nucleus division. After this, it leads to the cytokinesis. Cytokinesis means the cytoplasm division. This cytokinesis starts by the end of the anaphase or the beginning of the telophase. After the telophase, these two nucleuses formed, and at the center the invasion takes place, and the invasion starts developing longer and longer, and finally it divides both the nucleus, and finally the two daughter cells are formed. And these two daughter cells have all the same chromosomes which are present in the parental cell. So this mitosis is known as the equatorial division. Not equatorial, equational division, because equal distribution of chromosomes and all the materials will take place between these two daughter cells. In case of animals, this cell wall is completely absent. Because of this, the invasion takes place at the end of this outside the cell. But in case of plant cell, the cell wall is present. Because of this, after this telophase, the cytokinesis process starts at the center of the cell. This invasion starts at the center and grows outward to meet their extending the lateral walls. And the formation of new cell wall begins by the simple procedure called the cell plate, and that represents the middle lamella. In the plant cell, the cytokinesis starts at the center, but in the animal cell, because it doesn't have the cell wall, the invasion takes place from the outside. But in case of plant cell, the cell wall is present. Because of that, the cytokinesis distribution takes place from the center of the cell, and it moves towards the outer side, and finally the two daughter cells are formed.